Hi. Industrial fermentation media is composed of different components. An optimal fermentation media is essential for the growth of microorganisms as well as for the product formation. In this video, I am going to talk about how to formulate an industrial fermentation media. An industrial fermentation media should be a complete media. You should have components for all the metabolic activities of the cell, growth of the cell, as well as for the product formation. So the components should the requirements of the different components will vary depending on the type of microorganism, and so if the one type of microorganism may need these components in particular proportions, maybe a different kind of microorganism may need it in a different composition, different proportions, but more or less the requirements of components are more or less unchangeable. So it should have the media should have the components for biomass production. The microbial biomass is composed of the macronutrients and micronutrients. So it contains carbon, hydrogen, nitrogen, sulfur, phosphorus, etc. and some micronutrients. As I said, the proportion in which a particular microorganism is requiring these components will vary, but the basic requirements will be always the same. Secondly, it should have the components for the metabolite production. When we are using this fermentation for the primary metabolite production or secondary metabolite production, the media should have the components to support the metabolite production. Some cases, the media should have the precursors for the secondary metabolites so that the production will be easy. Thirdly, it should have energy. It should have the source of energy. In the most of the media, the carbon source itself act as the source of energy. If you are using carbohydrate as a carbon source, carbohydrate is also a source of energy. In some cases, nitrogen sources can be act as a energy source, but in case of phototropes, it will use light as an energy source. So some other microorganisms sometimes use the inorganic energy sources also. So the media should meet the energy requirements of the microorganism, and the media also should have components for the cell maintenance. So some microorganism may need some vitamins or some amino acids are essential components. So it also should be there in the media. So in a complete media, it should have components for the biomass production, metabolite production, energy, and cell maintenance. So if you want to put it in an equation, it goes like this. The microorganism use carbon as energy source along with the nitrogen source. Oxygen is in the form of aeration and other requirements. So our input is this much. We are giving them carbon and energy source, nitrogen sources, oxygen and other requirements. What microorganisms do by using all these components, they convert these components into biomass, the products, the primary or secondary metabolites, whatever may be the product. Then as a byproduct, it produces carbon dioxide, water and heat. So this is the basic equation of a fermentation process or the bioprocess. So it uses some components and creates some other components. The most important factor in a medium is carbon source. More than important, it used in huge quantities. Maybe 70 to 80% of media is composed of carbon source because the microorganism require this carbon for the biomass production, it require for energy, and it also require for the product formation. So microorganisms need this carbon source in high quantities. So and it has, as we require it in large amounts, huge quantities, it has a good impact on the production cost. If you are using an expensive carbon source, the production cost will also go up. And there is a relationship between the cost of the carbon source and the quality and the purity. If you are buying a cheap carbon source, very cheap carbon source, what happens is like that the carbon source is generally used to be they are impure, so purity won't be there, and expensive carbon sources used to have high purity, so they are related. If you are buying carbon source for very cheap price, it is purity will be so low, very low. And if you are getting the carbon sources for high price, it may have, well, in most cases it will have high purity and quality. And ease of sterilization and generalization of properties has an impact on on the fermentation. So our carbon source should be sterilizable, and gelatinization properties should be met for the fermentation. And it will require, as we need it continuously in large quantities for an industry, we should have a strategic plan to get this carbon source continuously for the industry. So if it's like a micronutrient or a precursor, in the most of cases we need it only in very small quantities, and we can also store in somewhere. But in case of carbon source, most of the bioprocess industry require it continuously and in huge quantities, and its storage is difficult. Even if we can store, it will add cost to the product. So we should plan there even before starting the industry. We should have a strategic plan to get this kind of carbon source continuously. In some cases, government legislations prevent the use of some carbon sources. For example, if you are starting a fermentation industry for the production of alcohol, maybe alcohol for the as a biofuel, the industry is making biofuel from some crops. Maybe you are using rice as a substrate, as a carbon source for the production of uh, alcohol. So you are taking the rice, buying the rice from the market, and your industry is converting it to fuel ethanol. But what happens is that if many industries are using rice as a substrate, carbon source, and making ethanol out of it, the price of the rice will go up, and the availability will be low, and it will cause so many social problems. So in order to prevent that, most of the live crop plants, crops like the cereals and wheat, rice, etc., cannot be used as a carbon source for industry, especially for the alcohol production and fuel production. We cannot use because government legislations prevent the use of such components, such commonly used uh, crops, yield, things like that in the for the production of fuel and alcohol. So carbon is required for the growth, cell maintenance, product formation, and it also provide energy for the microorganism. And one concept is like biomass yield coefficient. Biomass yield coefficient is like how much carbon source is utilized by the microorganism and how much biomass is created. So by consuming this much of carbon source, how much biomass is created. That's what the biomass yield coefficient is says. It's also called cellular yield coefficient. In by definition, it is the weight of an organism produced from a given weight of a carbon source. So suppose if you are feeding the microorganism with 1 kilogram of a carbon source, how much biomass is created? By using this carbon source, how much biomass is gained by the microorganism? That's what the biomass yield coefficient say. Its equation, it is says y carbon equal to biomass produced in grams divided by carbon substrate utilized. So for example, if you are cultivating bacteria under aerobic conditions generally and using glucose as a carbon source, its biomass yield coefficient is 0.5. That means if you are giving 1 kilogram of carbon source glucose to a bacteria, the increase in biomass will be 0.5 gram. If you are giving 100 kilogram of carbon source, you will get half of it. That is 50 kilogram. So it's like that. It's a yield is that much, 0.5. How much for a bacteria in under aerobic condition? Whatever the amount of carbon source you are providing, the biomass gain will be 0.5 percentage. Rest of the carbon source will be utilized for the energy, maintenance, product formation, etc. In case of Saccharomyces cerevisiae, if you are using the same glucose as a carbon source for Saccharomyces cerevisiae, at aerobic condition, the biomass yield coefficient is 0.56. And anaerobic condition, it is 0.12. As we know, there is a huge difference between aerobic respiration and anaerobic respiration. The yield of energy from aerobic respiration is very high because we have the glycolysis, then we have the Krebs cycle, and the NADP and NADH we are getting from the aerobic respiration. We can use in the electron transport chain to make more and more ATP compounds. But in anaerobic respiration, that thing is not happening. So the yield coefficient is so low. So if you are feeding the Saccharomyces cerevisiae with 100 kilogram of glucose, but you get at aerobic condition, the biomass yield is 56 kilogram. The yeast will grow into 56 kg, and if you are giving it in anaerobic condition, you will get only 12 kg when we are giving 100 kilogram. So that's what the biomass yield coefficient means. And there is different types of carbon sources are available, like it's wide variety of carbon sources are available for use, which includes carbohydrates, different carbohydrates. Maybe it has the monosaccharides, disaccharides, the trisaccharides, polysaccharides, etc. can be used as a carbon source. We can use glucose as a carbon source, which is a monosaccharide. Sucrose is a disaccharide, or lactose is a disaccharide. Then we can use starch. Maybe we can use cellulose, etc. can be used as a carbon source. So carbohydrates are very good carbon sources. Then we can use alcohol, alkenes or organic acids as carbon source. In some cases, we are using animal or plant oils and fats as carbon sources. Hydrocarbons are methane and or methanol, which is derived from petrochemicals, can be used as carbon source. In case of autotrophs, carbon dioxide is the carbon source. So I'm going to talk about each of these carbon sources, and the explanation will be very. I am not going to spend too much time in explaining all this. So one thing I want to tell before that is that there are two different types of carbon sources. One category is called readily metabolized sugars. Readily metabolized sugars are basically monosaccharides or disaccharides. Carbohydrates, like the carbon source like carbohydrates, the monosaccharides and disaccharides, like glucose or sucrose. And we do not really use this readily metabolizable sugars widely in industrial biotechnology. There are different reasons are there. One reason is that if you are using monosaccharides or disaccharides as carbon source, the bacteria or the microorganism, yeast or bacteria, whatever it may be, they will immediately absorb this carbohydrate into the cell. Inside the cell, it will immediately quickly undergo this Krebs cycle and glycolysis and Krebs cycle, and it will produce huge amount of energy. The carbohydrates, the carbon source doesn't need any processing by the bacteria. So the metabolic rate will be so rapid, microorganism will grow so fastly. But the thing is that in order to support that huge, very fast rate of metabolism, we should have enough oxygen supply. Otherwise, the microorganism will go to the anaerobic respiration. So oxygen supply should be very high, and the cooling of the fermenter, because the fermentation is an exothermic reaction. If it happens very quickly, temperature will build up in the fermenter, and which is not good for the microorganism. So we should have enough facilities to provide aeration, good aeration, and cooling. So one reason we are not using readily metabolized sugars are high demand for oxygen and cooling. Secondly, if you are making secondary metabolites, bacteria and fungus do not make secondary metabolites in presence of easily or rapidly utilizable sugars. If it has glucose in the medium, it won't make secondary metabolites. As we know, the primary metabolites are produced in the log phase of growth, and the secondary metabolites are produced in the stationary phase. If you are adding this monosaccharides into the medium, the bacteria will maintain in the log phase. It won't go to the stationary phase. Stationary phase is when there's nutrient limitations are there or toxin limitations are there. So if you are adding more and more this readily rapidly utilizable sugars, microorganism won't grow to the stationary phase of growth. Therefore, it won't produce secondary metabolites. So that's another reason. For example, if for the penicillin chrysogenum, if you add glucose into the medium of penicillin chrysogenum, it won't produce penicillin because penicillin is a secondary metabolite. If you are adding glucose into the medium, the fungus will be maintained in the log phase of growth. So in log phase, they won't produce the secondary metabolites. So that's why we are not using pure glucose or sucrose as carbon source in many cases. But in some cases, they are the most preferred carbon source also. For the production of primary metabolites, we can use glucose and sucrose. For the production of wine or for the production of alcohol, glucose and sucrose is used as the carbon source. But in generally, for the production of secondary metabolites or some other components, we don't use glucose and sucrose as carbon source. So that's all for this session. In this session, I will explain different carbon sources in detail. Thank you.